Welcome to the Deep Dive. We're here to take complex subjects, break them down, and give you the key insights you really need.
Helping you feel truly informed.
Exactly. And today, we're diving into something absolutely fundamental. Breathing. Since ancient times, it's been seen as synonymous with life itself, right?
Absolutely. Think of the biblical reference, God breathing life into Adam.
Right. But it's fascinating how the why was understood so differently. Like Hippocrates, way back, he actually thought breathing was mostly just to cool down the heart.
It's amazing, isn't it, how far we've come from those, well, let's call them intuitive beginnings.
For sure. And that journey of understanding is what we're tackling today. We're doing a deep dive into Chapter 26 of Boron and Boulpaep's Medical Physiology, the organization of the respiratory system.
It's a cornerstone of physiology.
Definitely. And look, this stuff can seem dense, but our mission here is to unpack it all step by step from the ground up. We want you to get a really clear, engaging picture, clinically relevant too, all without needing diagrams in front of you.
Finding those crucial nuggets of knowledge.
Exactly. So you walk away feeling confident and well-informed about how this incredibly vital system works. So let's start with the history, because it's actually pretty surprising. Go back to the 18th century, before oxygen was even a thing. Chemists had this idea of phlogiston.
Ah, yes, phlogiston, the fire essence.
Right. They thought air could only hold so much of this stuff, and once it was saturated, things couldn't burn anymore, and you couldn't breathe it. It seems almost bizarre to us now.
It does, but these early ideas, even the wrong ones, often paved the way. Phlogiston really set the stage for chemists like Joseph Black in the 1750s.
What did he find?
Well, he heated calcium carbonate limestone and found it produced a specific gas different from normal air. He called it fixed air.
Which we now know is CO2, carbon dioxide.
Precisely. And this was huge because it showed that air wasn't just one thing, but could contain distinct chemical gases involved in reactions. Then Henry Cavendish showed that fermentation and even putrefaction made this same fixed air.
Okay, so gases are chemicals. Then comes Joseph Priestley a bit later.
Yes, Priestley, in the late 1760s, early 1770s. He discovers another gas, which he calls dephlogistonated air. He noticed that burning things, decay, even breathing, consumed this air.
But plants produced it.
But green plants produced it. He even found a way to quantify it, reacting it with nitric oxide. It was a major breakthrough, co-discovered by Carl Scheele, too.
Now, here's where it clicks, right, with Antoine Lavoisier.
Exactly. Lavoisier, often called the father of modern chemistry. Priestley tells him about this dephlogistonated air, and Lavoisier connects the dots. He realizes it's not about air being full of phlogiston, it's about a specific element in the air.
And he names it.
Oxygen, O2. Lavoisier proposed that oxygen is consumed because it chemically reacts with substances. It's a chemical transformation, not just filling up the air.
A total paradigm shift.
Absolutely. And then, adding another layer, the mathematician Joseph-Louis Lagrange suggested something crucial. He thought, maybe this oxygen consumption and CO2 production isn't just happening in the lungs, maybe it's in the tissues themselves.
Moving beyond the lungs.
Right. And Lazare Spallanzani later demonstrated this rigorously in the late 18th century, showing that isolated tissues consume O2 and produce CO2.
So that leads us to the modern distinction.
Yes. We talk about external respiration, which is the whole process of getting O2 from the atmosphere to the mitochondria in your cells and CO2 going the other way. And then internal respiration or oxidative phosphorylation, which is the actual chemical process inside the mitochondria, using that O2 to make energy and CO2.
Got it. And for this deep dive, we're focusing on that big journey, external respiration.
Exactly. How we move those gases back and forth, and importantly, how that ties into maintaining your body's acid-base balance.
Okay, so with that history laid out, let's get into the mechanics. How do these gases actually move? The most basic way is diffusion.
Simple, random movement. Molecules just naturally spread out from where they're crowded, high concentration, high partial pressure, to where there's more space, low concentration.
And it doesn't require any energy from the body.
Nope, no energy needed. The driving force is simply that difference in concentration, that gradient.
Can you give us a picture of that?
Sure, imagine a tiny single-celled critter in a pond. Its mitochondria are constantly using up O2, so the O2 level inside is lower than the pond water. And they're making CO2, so the CO2 level inside is higher.
So O2 naturally flows in, CO2 flows out.
Exactly. It diffuses across these incredibly thin layers of water, both just outside and just inside the cell membrane. We call them unstirred layers.
And this follows a physical law.
Yes. Fick's law basically says the flow rate is proportional to that concentration difference. And thanks to Henry's law, we can relate concentration to partial pressure. So flow is proportional to the partial pressure difference, delta P. It works beautifully.
But only for really short distances.
Precisely. Once an organism gets bigger than about a millimeter across, simple diffusion just can't keep up. It's too slow to get oxygen to the core or CO2 out.
So nature needed another solution for us larger animals.
It did. And that solution is convection. Think of convection as bulk flow moving the fluid, air or water itself, to bring fresh supplies closer and wash waste away. This makes the diffusion gradients much steeper, speeding things up.
By stirring the pot.
Kind of, yeah. You see external convection even in simple organisms. A paramecium uses tiny hairs, cilia, to whip the water around itself. A clam actively pumps water over its gills.
But you mentioned water breathing is inefficient.
Incredibly so, because water holds so little oxygen compared to air. An oyster might pump 16 liters of water just to get 1 milliliter of O2. That's a huge energy cost.
Okay, so shifting to us mammals breathing air, that's our air pump doing the convection.
Exactly. Our chest wall, respiratory muscles, airways, that's our external convective system. The process is ventilation, moving air in and out, and it's way more efficient.
How much more?
Well, we might ventilate about 4 liters of air per minute to get 250 milliliters of O2. That's roughly a 16-to-1 ratio. Much, much better than the oyster's 16,000 to 1.
Because air just holds way more oxygen.
Right. But even so, we don't breathe as fast or as deep as we possibly could. There's a trade-off.
A balance.
Yes, a balance between keeping the oxygen levels in our lungs high and CO2 low versus the metabolic cost, the energy it takes to actually breathe. We settle at an alveolar PO2 around 100 millimeters of mercury and a PCO2 around 40.
And this balance is crucial. What happens if it's disrupted?
Good question. Clinically, think about barbiturate poisoning. These drugs can suppress the brain's respiratory control centers. Ventilation slows down or even stops.
So the air pump fails.
Right. The unstirred layer in the lungs effectively gets huge. Alveolar oxygen plummets, CO2 skyrockets. It's a critical failure of gas exchange, life-threatening.
Okay, so that's external convection, the air pump. What about moving gases inside the body?
That's internal convection, and it's handled by our circulatory system. Blood flow, or perfusion, especially through the lungs, is what brings the low O2, high CO2 blood right up to the air sacs for exchange.
And our four-chambered heart is key here, isn't it? With separate loops for the lungs and the body.
Absolutely essential. It maximizes those gradients. The right ventricle pumps that oxygen-poor blood only to the lungs. The left ventricle pumps the freshly oxygenated low CO2 blood out to all the body's tissues. No mixing.
Unlike, say, a fish or a frog.
Exactly. Fish and amphibians often have common heart chambers where oxygenated and deoxygenated blood can mix. That dampens the gradients, making gas exchange less efficient.
And we see problems like that in humans sometimes.
We do. Things like diseased heart valves that leak or congenital shunts holes between heart chambers, like septal defects, they cause that same kind of mixing. It impairs gas exchange because the blood going to the body isn't fully oxygenated and the blood going to the lungs isn't fully deoxygenated.
Okay, so we got diffusion, boosted by convection, both outside and inside. What else optimizes this whole process? You mentioned surface area earlier.
Right, surface area amplification. Fick's law also tells us that flow is proportional to the area of the barrier. More area means more diffusion.
So evolution found ways to pack in more surface.
Definitely. Aquatic animals often evolved gills, these intricate, folded structures sticking outwards to maximize contact with water.
And land animals went inwards.
Yes, with lungs. These are invaginations, internal structures. Amphibians might have relatively simple sacs, relying partly on their skin. Reptiles started getting more complex internal partitions.
And mammals took it to the extreme.
We really did. Our lungs are incredibly complex, with that extensive branching airway system leading to millions of tiny alveoli.
You mentioned earlier that our lung surface area is actually much larger than we need at rest, like 3 times faster.
That's right. It's an amazing amount of redundancy. And it's not waste, it's crucial reserve capacity.
For what situations?
For exercise, when your oxygen demand skyrockets. For high altitude, where the air has less oxygen. And even just for aging, as lung function naturally declines a bit over time, that reserve keeps us going.
So losing surface area is a big deal clinically.
It can be, yes. If someone has a lung removed surgically, they lose half their surface area. Or in pulmonary edema, where fluid builds up and thickens the barrier, that also impairs diffusion. Combine those, and it's very serious.
Okay, another fascinating piece you mentioned. Respiratory pigments. Why are these so important?
They're absolutely critical. Oxygen just doesn't dissolve well in watery fluids like blood plasma.
Yeah.
If we relied only on dissolved oxygen, our blood PO2 would quickly match the lung PO2, but we wouldn't be carrying enough total oxygen to survive.
Not even close.
Not even close. So evolution came up with these specialized proteins. Hemoglobin is the most common and efficient one in vertebrates, using iron in a porphyrin ring. But there are others, like hemocyanin, using copper in some invertebrates.
And hemoglobin's job is basically to grab onto oxygen.
Exactly. It binds oxygen reversibly. About 98% of the oxygen in your blood is actually bound to hemoglobin inside your red blood cells. This increases the oxygen carrying capacity enormously, roughly 65-fold compared to just saline or plasma. Plus, hemoglobin is also really important for carrying CO2 back to the lungs and acting as a crucial buffer to control blood pH. It's deeply tied into acid-base chemistry.
Which explains why anemia is such a problem.
Precisely. Anemia means less hemoglobin, so less carrying capacity for both O2 and CO2. The body tries to compensate. Tissues might extract a higher percentage of O2 from the blood passing through, or the heart might pump faster, increasing cardiac output.
But there are limits to that compensation.
Absolutely, which leads to fatigue, shortness of breath, and other symptoms.
So thinking about all these components, the pump, the surface area, the circulation, the pigments, what happens when one fails? You mentioned an interesting idea.
Right, it's this concept. Pathophysiology recapitulates phylogeny in reverse.
Meaning disease mirrors evolution backwards.
Kind of. When a key component of our sophisticated respiratory system fails, our overall gas exchange function can start to resemble that of a much lower or simpler life form. Okay, if the air pump fails, like in that barbiturate example, you're almost like a single-celled organism relying solely on diffusion in an unstirred environment. If you lose significant alveolar surface area, maybe it's like an amphibian with simpler sac-like lungs.
A major circulatory shunt.
That might mimic a fish with its mixed blood circulation. Severe anemia, it's like relying on a less efficient oxygen carrier, like some lower invertebrates. It's a powerful way to conceptualize the impact of respiratory disease.
That is a powerful framework. Okay, let's zoom right into the human system then. It truly seems like a masterpiece of optimizing all these aspects.
It really is. You can think of it as having 6 core, highly integrated components that make it all work so well.
Let's listen them out.
Okay. 1, the air pump itself, upper airways, trachea, bronchi, the chest cavity, the muscles of respiration. Inspiration is active muscles contract, chest expands, pressure drops, air flows in. Quiet expiration is usually passive muscles relax, lungs recoil. Gas carriage in the blood. That means red blood cells packed with hemoglobin, optimized for rapid loading and unloading of O2 and CO2, and that crucial role in acid-base balance we mentioned.
3 must be the exchange surface.
Exactly. The gas exchange surface, the alveoli. Huge area, incredibly thin, perfect for passive diffusion.
4, the delivery system.
The circulatory system. That 4-chambered heart, the separate pulmonary and systemic loops maximizing gradients.
5,
Local regulation. Clever feedback mechanisms within the lung ensure that airflow, ventilation, and blood flow, perfusion, are matched as closely as possible in different lung regions, even though they aren't perfectly uniform. Maximizes efficiency.
and finally the boss.
6, central regulation. Control centers in the brainstem rhythmically stimulate breathing muscles and adjust the rate and depth based on the body's needs, like during exercise. They get feedback from sensors monitoring blood O2, CO2, and pH.
An incredibly integrated system. Let's walk through the structures, maybe outside in.
Right.
What about the lining around the lungs?
That's the pleura. It's a thin, double-layered membrane. The outer layer, the parietal pleura, lines the inside of your chest wall. It produces a tiny amount of lubricating fluid, maybe 10 milliliter normally.
And the inner layer.
The visceral pleura, which is stuck directly onto the lung surface, it helps drain that fluid away via lymphatics. The fluid allows the lungs to slide smoothly during breathing.
What if too much fluid builds up?
That's a pleural effusion. It can compress the lung and make it hard to breathe. Also, just a note on lobes. The right lung usually has 3 lobes. The left has 2, leaving space for the heart. So the right lung handles slightly more of the workload, about 55%.
Okay, let's follow the air in, down the windpipe, the trachea, and then it branches.
It branches extensively, like an upside-down tree. The trachea is generation 0. It splits into the 2 main stem bronchi, generation 1. Fun fact,
Right.
the right main bronchus is a bit wider and more vertical, so inhaled foreign objects are more likely to end up there.
Good to know. And it keeps branching.
Yeah, about 23 generations in total in humans. And as you go deeper, the structure changes. Cartilage rings, which keep the larger airways open, gradually disappear. So do mucus-secreting cells and glands.
So the first branches are bronchi.
Right, up to about the 10th generation, they have cartilage and the mucociliary system, the mucociliary escalator, to trap and remove particles.
And after that?
They become bronchioles, starting around the 11th generation. No cartilage here. They rely on the surrounding lung tissue pulling on them, radial traction, to stay open, especially during exhalation when they might tend to collapse.
These early airways, bronchi down to the smallest bronchioles before the air sacs, they just move air, right? No gas exchange.
Correct. These are the conducting airways, from your nose down to the terminal bronchioles around the 16th generation. Their only job is bulk airflow convection. Their combined volume is the anatomical dead space you mentioned, air that doesn't participate in gas exchange. Maybe 150 milliliters in males, a bit less in females.
So air moves fast at first, then slows down.
Dramatically. The total cross-sectional area increases massively with each branching generation, so the forward velocity of the air plummets in the deeper airways.
And that's where gas exchange starts.
Yes, in the alveolar air spaces starting around generation 17. First, you have respiratory bronchioles, generation 17-19, which have some alveoli budding off them. Then alveolar ducts, 2022, basically tubes completely lined with alveoli. And finally, the alveolar sacs, generation 23, which are like clusters of grapes, the blind ends of the airways.
So down here, air movement is different.
Totally different. The forward velocity is practically zero. Gas movement over the last short distance across the thin barrier into the blood is dominated entirely by diffusion, not convection.
Got it. And the alveoli themselves, these are the key units.
Absolutely. The fundamental unit of gas exchange. Tiny little hemispheres, maybe 1/4 of a millimeter across. You have hundreds of millions of them.
Giving that huge surface area you mentioned, 50 to 100 square meters.
Exactly. An incredible amplification. And the lining is specialized. Mostly type I pneumocytes, very flat. Thin cells covering 90–95% of the surface, providing the shortest path for gas diffusion.
And type 2 cells.
Type 2 pneumocytes are chunkier, cuboidal cells, often found in corners. They produce that vital pulmonary surfactant, which reduces surface tension and makes the lungs easier to inflate. They also act like stem cells, repairing the lining if type I cells get damaged.
Fascinating. I read about pores of Kohn, too.
Ah, yes. Small holes connecting adjacent alveoli. Their exact function is still a bit debated, possibly collateral ventilation if an airway gets blocked.
And the blood supply is woven right in. You mentioned a dual supply.
That's right. The main one is the pulmonary artery system, coming from the right ventricle, carrying that deoxygenated blood. The arteries branch alongside the airways, eventually forming this incredibly dense capillary network that surrounds each alveolus like a mesh bag.
How long does a red blood cell spend there?
Not long at all, about 3/4 of a second at rest, but in that time, it might pass through the capillaries of up to 3 alveoli, efficiently exchanging gases. Then the oxygenated blood collects in the pulmonary veins to return to the left heart.
And the second supply?
The bronchial arteries. These are branches off the aorta carrying fully oxygenated systemic blood. Their job is to nourish the tissues of the conducting airways themselves, the bronchi and bronchioles, which don't get oxygen directly from the inhaled air.
Does that bronchial blood mix with the pulmonary blood?
Some of it does, yes. Where the systems meet, around the respiratory bronchioles, some bronchial capillary blood drains into the pulmonary veins. This creates a small, normal, physiological shunt or venous admixture, a tiny amount of less oxygenated blood mixing with the fully oxygenated blood heading back to the heart.
It's just so intricate. But the lungs aren't just for breathing, are they? You hinted at other roles.
Not at all. They're surprisingly multi-talented. Think about olfaction, your sense of smell. Ventilation brings odor molecules up to the olfactory epithelium in your nose. And that little sniff reflex, it lets you sample the air without taking a huge lungful of potentially nasty stuff.
Smart design. What else?
They're amazing at processing the air you inhale. Warming it up to body temperature is crucial for efficient gas exchange and prevents bubbles forming in the blood if cold air met warm blood. Moisturizing it prevents the delicate lung surfaces from drying out.
And filtering.
A huge filtering job. Breathing through your nose is particularly good. Nasal hairs trap, big stuff. The huge surface area warms and humidifies. Turbulence makes particles impact the walls, and that sharp turn at the back traps even more.
What about smaller particles that get past the nose?
The mucus and cilia in the trachea and bronchi trap most of those. Even smaller particles might settle out in the very slow-moving air deeper down. And the tiniest aerosols, less than half a micron, most actually get breathed back out.
But some still get deposited.
Yes, and the lungs have defenses. Immune cells, called macrophages, engulf particles. Enzymes break them down. Lymphatics carry material away. There's even a slow flow of fluid on the alveolar surface, moving particles towards the airways to meet the mucus escalator. And of course, coughing and sneezing are powerful reflexes to expel irritants.
Okay, air processing. Any other non-breathing jobs?
Yes, they act as a reservoir for the left ventricle. The pulmonary blood vessels are very stretchy and hold about half a liter of blood. This provides a buffer, ensuring the left heart keeps getting blood even if the input from the right heart flickers for a couple of beats.
Like a buffer tank.
Exactly. Another vital role, filtering small emboli from the blood. Your systemic veins can sometimes carry tiny blood clots, fat globules, air bubbles. The lung capillaries act like a sieve, trapping these before they can reach the brain or heart, where they could cause a stroke or heart attack.
So the lungs protect the rest of the body from these.
They do. The affected lung units can often recover, getting nutrients from those bronchial artery connections. But a warning, large or frequent emboli are obviously very dangerous, and unfortunately, the lungs can also inadvertently filter and trap cancer cells, allowing them to metastasize there.
Wow. Anything else?
One more key function, biochemical reactions. The cells lining the pulmonary capillaries, the endothelium, are chemically active. They selectively modify substances in the blood passing through.
Like what?
The classic example is converting angiotensin I into the much more potent angiotensin II using angiotensin-converting enzyme, or ACE. This is critical for blood pressure regulation. They also remove or inactivate other signaling molecules like certain prostaglandins, histamines, serotonin, bradykinin, while letting others pass through untouched. It's a sophisticated chemical processing plant.
It's incredible just how much the lungs do beyond simply moving air. So given all this complexity, how do we actually measure lung function? Clinically, how do we assess how well they're working?
That's where spirometry is fundamental. It's the measurement of the volumes of air you breathe in and out. Traditionally, you might imagine someone breathing into a bell floating in water connected to a pen drawing on a chart. Modern devices are electronic, but the principle is the same.
Just measuring air volumes.
Essentially, yes. Though we do need to correct the measurements because the air in your lungs is at body temperature and pressure, saturated with water vapor, PTPS, while the spirometer is at ambient temperature and pressure, saturated, ATPS.
Yeah.
A small but important correction.
Okay. And physiologists use specific terms for these measurements, right? Volumes and capacities.
Correct. There are 4 primary lung volumes which don't overlap. First is tidal volume, VT. The amount of air in a normal, quiet breath, maybe 500 milliliters or so.
Like the tide coming in and out.
Exactly. Then there's inspiratory reserve volume, IRV, the extra air you can breathe in with maximum effort after a normal inhalation.
And the opposite for exhaling.
Yes, expiratory reserve volume, ERV, the extra air you can force out after a normal exhalation.
And the fourth volume.
That's residual volume, RV. This is the air that always remains in your lungs, no matter how hard you try to exhale completely. You simply cannot empty your lungs entirely.
And because it never comes out, you can't measure RV with a simple spirometer.
Correct. It stays trapped inside.
Which raises the question, why have it? Why not just empty the lungs completely? Seems inefficient.
But it's actually very clever design. 2 main reasons. One, it helps prevent airway collapse. The smallest airways lack cartilage and tend to snap shut. Keeping some air in them prevents this, reducing the effort needed to reopen them with every breath. It saves energy.
Makes sense. And the second reason?
It allows for continuous gas exchange. Remember, blood flow through the lungs is constant, but breathing is episodic. That RV acts as a buffer, mixing with the fresh air, so the oxygen and CO2 levels in the alveolar air don't swing wildly up and down with each breath. This keeps blood gas levels much more stable.
Okay, so those are the 4 volumes. What about capacities?
Lung capacities are just combinations of 2 or more volumes. Total lung capacity, TLC, is everything, all 4 volumes added together, the absolute maximum air your lungs can hold.
What about after a normal breath out?
The air left then is the functional residual capacity, FRC. That's the ERV plus the RV. It's the resting volume of your lungs when your breathing muscles are relaxed.
And FRC includes RV, so you can't measure it with spirometry alone either.
Right. Then there's inspiratory capacity, IC, which is your tidal volume plus your inspiratory reserve volume, the most you can breathe in, starting from that FRC point. And finally, vital capacity, VC.
That sounds important.
It is. VC is the total amount of air you can move your IRV plus TV plus ERV. Basically, the biggest possible breath you can take in and then exhale fully. It's often monitored clinically because it changes with many lung diseases.
There's one other measurement often mentioned, FEV1.
Yes, forced expiratory volume in 1 second, FEV1. This measures how much of your vital capacity you can forcefully exhale in the first second. In healthy lungs, you should be able to get about 80% of your VC out in that first second.
Why is that useful?
It tells you not just about the volume of the lungs related to VC, but also about how easily air flows out, which reflects airway resistance. So it's really valuable for diagnosing and tracking conditions like asthma or COPD, where airways might be narrowed.
Okay, so if spirometry can measure TD, IRV, ERV, VC, and FEV1, but not RV, FRC, or TLC, how do we measure those volumes that include the trapped air?
Great question. We need different techniques, usually based on the principle of conservation of mass, basically, tracking a known amount of gas. One classic method is the helium dilution technique.
How's that work?
You have the person breathe from a spirometer that has a known volume and a known concentration of helium gas. Helium is used because it's poorly soluble. It won't really leave the air and dissolve into the blood.
So it just mixes with the air already in the lungs.
Exactly. As the person rebreathes, the helium spreads out into their lung volume. The concentration of helium in the spirometer goes down. By comparing the initial and final helium concentrations and knowing the initial spirometer volume, you can calculate the unknown lung volume the helium diluted into.
Clever. So if you start the test after a normal exhale, you measure FRC.
Precisely. Or if you start after a maximal exhale, you measure RV.
What's another way?
The nitrogen washout method. This time, you start with the person breathing normal air, which is about 75–80% nitrogen. Then you have them switch to breathing 100% pure oxygen.
Washing out the nitrogen.
Exactly. All the nitrogen exhaled is collected in a large bag or measured by flow meters. Since you know the initial nitrogen concentration in the lungs, around 75%, and you measure the total volume of nitrogen washed out, you can calculate the lung volume it came from. Again, FRC or RV, depending on when you start.
Okay, one more method, the body box.
Yes, body plethysmography. This one uses Boyle's law. Remember, P times V is constant if temperature and amount of gas are fixed. The person sits inside an airtight box, like a small phone booth.
And then what?
They make a small breathing effort, like trying to inhale against a closed shutter or valve. When they try to inhale, their chest expands slightly, increasing their lung volume, delta V. This increased volume decreases the pressure inside their lungs, delta P, according to Boyle's law.
And the box measures the volume change.
Yes, the box is sealed, so as the person's chest expands, the air volume in the box decreases slightly, and the box measures this tiny change in volume, which equals the change in lung volume, delta V. By knowing the initial pressure, atmospheric pressure, and measuring the change in pressure in the airway, delta P, and the change in volume, delta V, you can calculate the absolute volume of gas in the lungs, V, when the shutter was closed.
Also measuring FRC or RV.
Exactly. It's often considered the most accurate method as it measures all the gas in the thorax, even gas trapped behind blocked airways, which the dilution washout methods might miss. #tag outro.
Wow. Okay, that was quite a journey. From thinking breathing just cooled the heart, through phlogiston, to discovering oxygen, understanding diffusion and convection, the incredible surface area, the vital role of hemoglobin, all those non-respiratory functions. And finally, how we actually measure these volumes.
It really highlights the elegance and complexity of the system, doesn't it? How physics, chemistry, and anatomy all come together. Understanding these connections, from the cellular level up to the whole organism, is really the key to grasping physiology and why it matters clinically.
Absolutely. Hopefully this deep dive helps you, our listener, feel a bit more confident navigating these concepts. It's not just about facts and figures, it's about appreciating this incredible biological design.
You really are capable of mastering this material. Remember that.
Definitely. So here's a final thought to leave you with.
Given everything we've discussed, the built-in redundancy, the multiple functions like filtering and biochemical processing, what do you think might be the next big leap in understanding lung adaptation? How might they respond to, say, extreme environments like space travel, or how could we better harness their capabilities to tackle new medical challenges in the future? Something to ponder.